I have a lot to get through today, as you can maybe imagine from the title alone being a bit of a mouthful. So I'm not going to spare too much time with any idle introductions. Just quickly, my name is Erik. I work part-time at the Natural History Museum of the University of Oslo and part-time at CICERO, a climate research institute in Oslo. I will soon be making the move to work 100% at the Climate Research Institute. So if you see any emails under which you can reach me on the slides, you will find those are already of the new workplace. So if you have any questions going forward, I would ask you to contact me there. With all that being said, I work adjacent to GBIF in Norway, and I work extensively with climate data, which is why I would like to today talk about how to investigate biodiversity data using open access databases, following fair data principles, both regarding biodiversity data and environmental data. Both of these are highly important data sources for your work as biologists, ecologists, you name it. However, both are usually poorly understood by your average ecologist. That includes myself from a few years ago. So this is not just me preaching from a podium. I clawed my way up here. So let's get started with talking about biodiversity data. Basically, myself as a biostatistician, how can I learn about the outdoors and by extension you learn about the outdoors without needing to actually step foot in them? As much as we like them, sometimes it's just easier to retrieve someone else's data and run our analysis on those. Well, the answer to that is for most people, GBIF. However, first we need to understand what GBIF even is. GBIF is an intergovernmental network research infrastructure, which makes available to anyone everywhere, as long as they have an internet connection, freely available open access data about any type of life on Earth. There are a few caveats to this, which we'll get to in just a second. However, for now, just know the mission statement of GBIF is to make information about life on Earth available to everyone. GBIF is massive. As you can see here, there are over 3 billion data points in there as of last month, supported by over 100,000 data sets that contain those data points, with support for more than 11,000 peer-reviewed papers using and citing GBIF data. There is a large number of papers that use GBIF data without actually giving proper credit, which is something that we'll get to today. So there is a large number of papers that should show up here that aren't there. Monthly, there have been over 154 billion downloads in the year 2022 from GBIF. This is downloads, not observations being downloaded. So you can multiply these 154 billion per month by probably a few thousand to a few 100,000 of records contained per download. It is a massive undertaking, GBIF. And we need to understand what data is in it so that you can understand how you can make the best use of it. GBIF, in the shortest form, tells us about evidence about where species have lived and when on our planet. This comes down to digitized specimens, such as museum collections that have been digitized, observations being made in the field, literature reviews telling us where people have claimed to have cited certain species, remote sensing, and also environmental DNA sequencing, all of which are very different data types that get harmonized via common standards. In the context of GBIF, this is the Darwin core. So if you ever see this, just know that this is a metadata standard by which to collect information about vastly different observation types so that we can integrate them with one another. These then get aggregated and indexed by GBIF so that you can interact with them. So there's a lot of data in there. How do you get to use it? Well, first of all, there's a lot of use cases that GBIF does support. Firstly, not only does the data come from a lot of different sources funneling into GBIF, GBIF then also funnels that information back out to a lot of different use cases supporting both research initiatives as well as policy making. You might find yourself on the left-hand side of the spectrum or on the right-hand side of the spectrum and in different bubbles. So now we need to understand how we can access GBIF. And this is where I need to take a quick break and tell you guys that I have prepared training material for this course that you can find on my website. I'll show you later where to find it. I do not expect you to do any actual hands-on coding today because I've got a lot of material to go through. However, I will show you excerpts of that practical material so that you can see what you would be doing if we did have the time to cover it all with some free flow work. If we end up with some time at the end, I'm happy for you to boot up your R sessions and actually do some of this work. However, for now, I would just like you to know that you can sit back, relax. All of what you're about to be shown is also available online for you to interact with. This is the pipeline for how to get data from GBIF. I am saving you all of the talk about how to interact with GBIF through the website or the web portal of GBIF, because quite frankly, I think it's a suboptimal way of interacting with GBIF data, particularly if you want to author reproducible research. So we're just glancing over that right away. And we are stepping into programmatic retrieval of data mediated through GBIF. This starts with first discovering the data we're interested in, then downloading that data, processing that data, and finally, when we're ready with our analysis, properly accrediting the data. So let's go through those step by step. With the example of pine trees and deciduous trees, Pinaceae and Fagaceae, I like both of those tree groups because I think they're really pretty whenever I go out in nature. However, I also didn't want to limit ourselves to a single species because then I would have to have some good justification for that. So we're just running with trees today. Firstly, we need to understand how data is indexed within GBIF, because Pinaceae and Fagaceae are very robust names of entire taxonomic groups, which are likely not going to change in the near future. However, as new species are being described and new evidence suggests that species membership in different taxonomic families, groups, you name it, should change, so will their names. So while the binomial nomenclature that we usually work with when we talk to each other about species is really useful for us as humans to understand it, it is not entirely practical for machines and for use of data in perpetuity. So GBIF indexes data differently. While you still get access to the binomial nomenclature, GBIF uses persistent unique identifiers, basically assigning numbers to each species identity instead of names, with a separate lookup table for what each different number has as the clear name, or as the binomial name, and then different clear names in different languages. The idea here is that persistent unique identifiers are, as the name suggests, unique to each species and persistent in that they don't change when species names or species grouping changes. These are, as I said, unique, persistent, and most importantly, resolvable by machines. And you need to interact with them, as GBIF has those kinds of identifiers all over itself. So understanding how to use these identifiers is crucial for obtaining the data that you actually need for your specific research purposes. And this is how you do it. You have a particular species, or in our case, taxonomic group in mind. You use the R function from the RGBIF package, name_backbone, where you just put in the name that you're after. On the left-hand side, you see Pinaceae. On the right-hand side, you see Fagaceae. And ultimately, from that, you get a data frame where you can then extract, in our case, a family key, because we're looking at taxonomic families, or a species key if you're looking at species, which then gives you the persistent unique identifier of those specific groups. In our case, 3925 for Pinaceae and 4689 for Fagaceae. For species, these keys will be a lot longer because there's a lot more species. These are numbered from top of the taxonomic rank down to the lower rungs like species, with Animalia and Plantae, I think, taking 1 or 2 respectively. So now we have our persistent identifiers. How do we find data within GBIF or understand what information GBIF makes available to us regarding these two families? There are 2 functions that you could use, and I will strictly advocate against the first, which is OCC_search. You will see this crop up, and I want to tell you about it because I don't want you to use it. as OCC search can give you up to 100,000 records matching a query from GBIF right out the gate. Sounds great. 100,000 data points is a lot of information. However, for especially families of entire groups, we get way more than 100,000 records within GBIF. Meaning if you download only 100,000, you will somehow, and very non-understandably, bias your knowledge of these groups. Meaning, please do not use this function. It gives you more functionality than the next function I'm about to introduce. However, it doesn't give you enough functionality to fully use GBIF effectively, which we will use separate download staging for in just a second. Sorry, a question in the chat I'm just going to respond to right now. Are family name keys different from species keys? E.g., is there a 3925 species as well, or are species keys always longer than 4 digits? These keys are not separate by family or species or other taxonomic hierarchies. There is no species that is 3925. A Pinacea species will have a completely different number. It doesn't even start with 3925. If so, it would be by chance. Species numbers are usually longer. Which then brings us, returning to the presentation itself, to the function that I actually would like you to use, which is OCC count. OCC count just tells you how many records are within GBIF that match your query. Very easy, because now you know how many records do we have. Does it make sense to maybe use OCC search? It doesn't. Never use OCC search. What we do here is ask GBIF, I'll walk you through the syntax of this, to please give us everything, and I see a lot of you taking, or some of you taking frantic notes. All of this is explained also on the website. Feel free to, you can take notes however much you want, but you can just lean back and enjoy the show, so to say. Not trying to tell you how to live your life, just wanted to stress that one more time. The syntax of the OCC count function has several different arguments. I'm just showing you the ones that are relevant to what I want to walk you through today. Firstly, we ask for every observation that is mediated by GBIF where the family key matches either the Fagaceae family key that we've already obtained up here or the Pinaceae family key that we've already obtained up here. We then ask for observations only within Germany. This seasonal school is hosted in Germany, so I figured this would be a good reference area to look at. And we're looking for the years 1970 to 2020, including both the start and the end year. And you can see that GBIF makes available almost 500,000 records for this query, showing you right away that having used the, I'm sorry, I really have beef with OCC search, which shows you right away that if you had used OCC search, you would have been entirely misled on the information that would be available to what you ultimately want to research. You can refine these searches more, looking for different quality flags in the data, looking for different contents in the data. All of this is explained in the help documentation of our GBIF and a bit more on the on my website where I have the actual workshop material for this. But now we know what data we want to query. We know how much data there is. It is now time to actually ask for that data. To ask for that data, we simply make a data request with the OCC_download function. OCC standing for occurrence, by the way, because this is the most common data type that is within GBIF and what you will most likely use GBIF mediated data for. With this function, we register a data request or query with GBIF. Unfortunately, the syntax for this function is slightly different to OCC count. However, you can query for the exact same things. And in this case, we're actually querying for a bit more because you will interact with this kind of function much more throughout your career within ecology than you will with OCC count. Because knowing how many observations there are is not quite as effective as getting all of them. What we do here is, again, just asking for all of the records that either match the Pinaceae key or the Fagaceae key. We ask for everything that has been recorded by a human because I want to exclude digitized specimens that are somewhere in some collections in museums because I only want actual individuals that are out in the wild. Then we still ask for Germany as the country, and we ask for years greater or equal than 1970 and lesser or equal to 2020, giving us almost the exact same amount of data that we just saw with OCC count, only this time we have added the basis of record just to show you that there are more arguments that we can pull in. Again, this can be refined further with additional arguments that, again, I explain more on the website version of this. So now we've made our download request. We then have to wait for that download request to be fulfilled by GBIF. GBIF has to now sift through literal billions of records and return the ones that match our query back to us. GBIF is very efficient at this, but GBIF also gets pinged for a lot of downloads on a monthly basis, as you've seen previously. Therefore, particularly when we request either complex or very large data sets, this can take some time. And if we just use the OCC_download_wait function, we just have a small function that keeps our R session busy and just every, in this case, 10 seconds right here, pings the server and just asks, are you done yet? Are you done yet? Are you done yet? And when it's actually done, the session will progress. So we're having a nagging child on the back seat here that annoys the GBIF servers until they're ready, but we don't have to do it manually. And GBIF doesn't get upset with you doing this. GBIF has authored this R package. Once the download is actually ready for us, we need to make the actual download. For this, we use the OCC_download_get function, which then downloads a zip archive onto our disk corresponding to the download request that we made in the first place. This zip file will have have as a name the ID of the query that you have lodged with GBIF itself, which is not what you will end up citing. This is an important distinction which a few people get wrong sometimes. Finally, to get the data that is contained within the zip file into R, you just use the OCC_download_import function onto the output of the OCC_download_get function, and then you get a massive table loaded into your R session, much bigger than what I'm showing over there. And then you have your data. Now, of course, sometimes you will want to throw data away again as your hard drive gets clogged up, as you move on to other projects and archive things. I would usually recommend that you archive all of the raw data that you've downloaded on an external hard drive. Just zip it up some more or zip it up again if you've de-zipped it at some point, and just have it rot away on some old hard drive that collects dust in the back of your shelves, because nothing beats an actual physical backup. However, GBIF tries to store downloads, so the finished query that you have made, for as long as possible. But even GBIF works with only limited disk space. Granted, much more disk space than you or I will ever have in our respective laptops or living rooms. However, still limited disk space. So they can't store resolved queries in perpetuity. What they will prioritize in storing resolved queries are those that are actually cited in peer-reviewed research. In addition to that, you get for each of your downloads that you stage through the OCC download function, not through OCC search, another big misstep in using OCC search, you will get a DOI that you can actually cite. And this DOI will always, on the back end of GBIF, contain the specification of your query at the time of you making it, so that you can rerun the query from the data that GBIF hosts, but you might not get the exact same data back as the data backend catalog changes over time. So the recipe is stored, not the meal. So now we've talked about you needing to accredit GBIF data and the download that you've made in order for it to continue to be stored and for you to be able to look back on the recipe in case you have lost or, worse yet, not managed your scripts well enough to be able to return to them. The way you cite data that is mediated by GBIF is to first download data from GBIF, otherwise you've got nothing to cite. Then you receive the recommended citation with a download DOI, which is not the ID of the downloaded zip file. Big difference there. And then finally, you just cite the DOI and publish research or other work. How do you now get this DOI, you will rightfully ask? You can either go onto the download page on your GBIF user account and retrieve it from there. However, if you stage a lot of downloads, that list will fill up and you will not quite know what links to what without doing too many mental gymnastics. Luckily, you can do this directly in R as well. If you just call the OCC_download_meta function and then just subset for the DOI field in there, you get the DOI of the process data request from GBIF that you can then use to cite like such for the actual data that I've downloaded for this course. And that should be available somewhere in an already cleaned and handled state on the back end for you in some repository, if I'm not mistaken. So now you know how to accredit GBIF data. What is the benefit of accrediting GBIF data? We've already said that if you do so, your download request that has been processed and is finished will be stored for longer. You can go back to the download specification as hosted on the GBIF server with that DOI. However, let's say that you're the one actually publishing data, something that I will not be covering today in too great a detail. However, I want to sensitize you to how much you as a data curator can benefit from this DOI system and how much you as a data user are obligated to give proper credit. Because as soon as you cite the DOI of your download right up there, it then resolves to the involved data sets, which have their own DOIs, in this case here a floristic record from the Bayerische Landesamt für Umwelt. And ultimately, then, you can track the citations of these data sets, and more granularly, this resolves back to the contributors of this data set. So if you have contributed to this particular data set, and we were to publish something containing this download right here that we've made for this course, you would be given credit. Granted, the way academia is set up, that credit is not worth very much, but this is a systemic issue within academia that values peer-reviewed papers over everything else, and that's a longer discussion that I'm happy to rant over in a different occasion on how the system's broken. I think I'm preaching to the choir, so I'm just going to move on from that. Just note that it is in your best interest to cite the DOI because you want others to cite the DOI as well. This is just the summary of what I just said. And so now we have data, and we want to handle or process this data to extract the contents that we are either A, interested in, B, can use, or C, we trust. With managing billions of records, GBIF cannot guarantee that the data is unbiased, 100% correct. However, it does its best job to tell you about potential issues. The very first issue that you need to be aware of when using GBIF in particular is that birders are crazy people. I have big respect for them. However, birders really like what they do, and I'm very happy for them. I wish I could be as passionate about something as birders are about contributing to GBIF, because you will immediately see on this diagram on the right-hand side here that compared to the biodiversity that we expect to be out there of birds, we have a vast overrepresentation of them within GBIF. Almost a third of GBIF is just birds. All life on Earth is not 1/3 birds, as much as birders would probably like it. And so be aware that when you obtain information from GBIF, particularly when spanning across the tree of life, you will get a strong bias for some groups over others, because it seems no one likes to observe insects and tell GBIF about it, at least relative to the biodiversity of insects that we expect on Earth. So if you wanted to do a comparative study of birds and insects, because you are interested in how birds feed on insects, for example, GBIF will not give you an evenly spaced picture of life on Earth, considering these two groups on the most extreme ends of the spectrum. There's nothing we can do about this other than trying to start a movement to get birders, but for insects. It is just important for you to be aware that there might be biases. There are more biases, however, not just taxonomic ones. There are also spatial biases. This right here is where each year we hold an open science course for biologists. Little plug, it's really nice. It's a really good course. It's probably running again next year. This always happens at the beginning of winter, so you're likely to get a bit of snow in the beautiful national park scenery of Dovrefjell here in Norway, right there at a really nice little hotel. What I want to point out with this, though, is that the charismatic species that this area is known for, the muskoxen, is, when asking GBIF, only observed right here on this left-hand side. There's a train line and a road that seems to bifurcate where they're observed. Now, these creatures are massive. The road isn't very much used, and the rail runs maybe 3 times a day. There's no good reason to believe that this is actually as much of a dispersal barrier as we would believe from GBIF itself. So why then is there this spatial bias? Well, because observations are driven by where people go and by visibility. So on the left-hand side here in the west, we have a national park. People usually hike in national parks, especially Norwegians. So we would probably get more opportunistic observations over here from hikers, where people go. On the right-hand side of the trail, of the rail line and of the road in this case, we have a mostly forested area, whereas this over here is exclusively tundra, meaning that when you sit on the train or in a car, you can see muskoxen out to your left, but not out to your right when going northwards. Meaning we have factors that compound on one another. Surely there is some dispersal barrier being posed here by the road and the rail line. However, it is definitely not as starkly contrasted as we would believe from GBIF. Again, finding such spatial bias and actually being able to attribute it is very difficult. And if anyone is interested in a master's thesis project, we are engaging with exactly this. We are advertising one right now at the Natural History Museum here in Oslo. I'm sorry, I'm just shamelessly plugging today. Sorry for all of the organizers that I'm just doing this. We also have temporal biases within GBIF. You'll see here that in this 5-year window on Darwin's voyage, we do actually have data from back then that is available through GBIF. However, you can really see that only Great Britain was really observed and the Netherlands, because everyone was into their orchids at the time. If we now use a 5-year time window for present day, that story looks a bit different. Obviously, we have more people on Earth, we have better connected people, we have more money being invested into research, well, not relatively to GDP, but nevertheless, we do have more research going on in total. And ultimately, then we get a lot more information about life on Earth. And if you graph this out, you see an almost exponential increase of records mediated through GBIF as time goes on. Which, if you want to compare, for example, species distributions of certain species in the 70s to the 2010s, you will most likely run into the issue that you have very different sampling densities and sampling efforts expended at both of those time intervals. Again, be aware of those. There's probably not much you can do about them. Just be aware of them. Know the limitations of the data that you have so that you can accurately convey the limitations of the findings that you ultimately arrive at. All of this, to an extent at least, GBIF tries to tell you about with data issues and quality flags that you can further subset your downloads by or process your data with, which I'll show you in just a second. These are automatically applied by GBIF, and you will probably want to do a bit more data handling on your own end to throw out suspicious records. For example, on the right-hand side, you can see an overview of all bryophytes, mosses, that GBIF tells you about. And there seem to be a lot of mosses right here off the coast of Africa, where there's no island. Does anyone have an idea why that spot is relevant? And it is relevant not just to mosses. This happens for every single group in GBIF. It's because that's where 0, 0 is. When you don't know what to type for your coordinates, you shouldn't type anything. Some people type 0, 0. Some people have scripts that automatically assign 0, 0, which is an incredibly bad idea, which leads to the fact that many people who are actually interested in this gulf right here struggle with having to subset for everything directly at 0, 0, even those data points that were actually observed at 0, 0. It's a real hassle. Be aware of this. Luckily, with what we work with today, for the sake of this workshop, we don't run into these issues because we are looking at Germany. And so let's actually step back into R and look at some of the quality flags. The first one that we want to look at is, out of all of the records that we downloaded, how many of those don't have coordinates associated with them? Let's assume that what we want to do with the data that we've downloaded is to run a species distribution model. We need coordinates for each observation to run those models. 480 out of our almost over 400,000 that we've downloaded don't have coordinates. We throw them out. We can't save them. And even if we could, it's probably not worth the effort with those few out of that many. Next, we look whether there are geospatial issues. That is a whole suite of potential biases or factors that might make the data unreliable. Are there any of those within our remaining dataset? Yes, 6 of them. We chug them out. Now there's an interesting one. Coordinate uncertainty in meters. I want to know only about trees belonging to Pinaceae and Fagaceae across Germany, that have less than a kilometer of observation uncertainty in their geolocation. That drastically reduces our data set from over 400,000 records down to just almost 60,000 records. Why do I want this? Because, and you'll see this also on the workshop material on the website, there are some entered that have coordinate uncertainty in the realm of 50 kilometers. Some have coordinate uncertainties entered that are greater than the circumference of the Earth, which is clearly something going wrong there. If we're not certain of where the tree actually is, can we reliably use it for species distribution modeling by matching it with environmental data? I would argue no. And so I'd much rather not use those data points. However, again, we could have a philosophical debate. Others would argue differently. I have a very conservative approach when it comes to using data and the quality therein. Finally then, and this is a very important one, so I want to spend just a minute talking about this. We have talked about occurrences. GBIF calls records or observations occurrences. However, we understand occurrence as observation of something being there. That is not how GBIF understands occurrence. Occurrence just means a record pertaining to a species as far as GBIF is concerned. That can mean that the occurrence status is present or absent. And we definitely don't want to feed our species distribution model with absent occurrences. We only want to use present ones. If we now look at our remaining almost 60,000 data points, we find that all of them are present, none of them are absent. Great, we don't need to discard any more data points. This will usually be the case. GBIF is dominated by occurrences that record the presence of something rather than record the absence of something. However, a few of those are in there, and it is very important for you to be aware of this distinction. Lastly then, and something we luckily don't have to deal with in the dataset that we are working with today, however, I want you to be aware of, is that taxonomy in its own right is an art and a science. And there are taxonomic homonyms where, for example, this bivalve on the left-hand side has the same name as that flower on the right-hand side. These have different persistent unique identifiers within GBIF. However, you have to be very careful when matching these names with the GBIF backbone to see whether you are actually looking at the right persistent identifier. The way you would do this for these two species in particular is that you would check which family and which kingdom they lie in, because then you can quite easily tell apart which one is the persistent unique identifier for a plant and which one isn't. And so finally, we step back into R and we transfer the big table that we had before, now that we've subset it all, into a spatial object. We assign regular Mercator projection, EPSG 4326, I think the short version is, and go along with it. This will be relevant for everything I show you subsequently, and I just want you to see how transferring a table into a spatial point object looks like, so that you're aware of those, because they are highly relevant for actually matching GBIF observations with environmental data. So let's take a quick detour into visualizing this data. Again, this gets expanded much more in the workshop material on the website. And having this spatial object, we can quite easily plot all of our Fagaceae deciduous trees in orange on the left-hand side, with the Pinaceae in green on the left-hand side, and their distributions across Germany. We can also aggregate by the different states within Germany, which shows us quite distinct distributions of Fagaceae and Pinaceae as understood through GBIF-mediated data. We can also graph out how our information about these different families has increased over time. And you can see on the left-hand side for Fagaceae, on the right-hand side for Pinaceae, what I mentioned previously, this temporal bias of an almost exponential increase in records available per family as time goes on. And with that, I would like to conclude the biodiversity section of this talk and quickly ask if there are any questions relating to the biodiversity part. Because right now would be a good time to take those before I move on into preaching about environmental data. What happens to PIDs if a species splits up taxonomically? That is a really good question that I actually don't know the answer to. I will have to ask our honchos that actually work for GBIF in the office just over there after the presentation. I'll make a note of it and I will get back to you over the course of the seasonal school. I would imagine that this is just me spitballing, but I would imagine that the PID for the previously merged species is retired, and 2 new PIDs assuming there's a split into 2 new species, are introduced. But I'll double check. All right, and with the polite question out of the way, so that one question was asked, I will then move on to talking about environmental data and justify the statement that I've put up on the slide here, where I believe that contemporary practices in ecology relating to environmental data, its retrieval, its handling, and its use are severely lacking. I'm leaving out the severely because I'm publishing these slides online and I don't want to upset too many people. First of all, we need to understand environmental data types. I don't know how many of you have worked with environmental data before, if you've collected environmental data in the field, if you've retrieved it for some analyses. I see a few nods, so I assume there's some base knowledge here. For everyone not initiated, there are different data sources of climate data or environmental data. I'll use those 2 words interchangeably because that's what I work with. You can use data loggers, which are single location measurements, as you can see on the right-hand side here, of a weather station up in Dovrefjell, where that course is held, nudge, nudge, which is a really good waypoint when you're stuck in a snowstorm, by the way, because this kind of shows you the way to the nearest cabin. You also get, at greater coverage, climate models, which model land, ice, ocean, and atmosphere, and look something like this, with gridded surfaces and models running across those gridded surfaces for the entire Earth or just certain areas of Earth, in case you have either a global climate model, a GCM, or a regional climate model, an RCM. You also get gridded observation products. These are single location measurements, like this weather station here, that are then aggregated together with other single location measurements and interpolated to fit a grid, much like the grids that climate models use. Finally, then you also get climate reanalyses, which are a synthesis of the climate models and the measurements on ground, however, not interpolating to fit some grid. How do we use these different data sources or these different data types? You will most likely have only interacted at macro scale, at least, with gridded observations. I would like you to stop that after today, and you'll see why in just a second. When we want to look at past and present conditions, we can use everything: climate reanalyses, gridded observations, and climate models. However, when we look at the future conditions, we can only use climate models. So then the question arises, why don't we use climate models for everything? Wouldn't that be the most consistent, not mixing apples and oranges? Well, in ecological models, what we do use is, for past and current species distributions, as an example, gridded observation products and climate reanalyses, and for future species distributions, this applies to other ecological studies as well. I'm just doing this with a species distribution modeling framework because I know that's what part of this seasonal school is built around. For those, we're using climate models for the future ones. The reason for that being is that the spatial resolution of climate models is the coarsest, i.e. the biggest cells with the least variation that we can understand to be linked to topographic features, for example, with climate reanalyses usually being in between and gridded observations giving us really high resolution data. On the temporal resolution side, we find that climate reanalyses actually give us the finest resolution, with climate models giving us the least resolution, or the coarsest again, annual averages, or even 100-year averages, and gridded observations giving us something in between. So you can see there are trade-offs to picking your data product. And I'd quickly like to tell you about the data products that are the most relevant to you, which are gridded observations and climate reanalyses. So what is a gridded observation product? Well, like I said previously, it's an interpolated single location measurement raster in the end. The process here is to obtain single location data from, for example, weather stations, and then use an interpolation algorithm to fill a predefined raster or grid. This has a few issues. First of all, you are limited in coverage, both in space as to where stations are, in time as to when stations were established and when they went defunct, and ultimately also in essential climate variables, ECVs, measured at those stations. That's terrific if you have one station that is kitted out with absolutely everything, but if no other station has it, then you can't interpolate. In addition to that, these data sets are subject to biases. These can be systematic, such that stations are placed non-homogeneously across Earth, which we know to be the case, as you can see. By the way, can you see my cursor? Fab, thank you. As you can see on the right-hand side here, these are weather stations used for the WorldClim product. And you can see these are very much not representing all spaces on Earth equally. Also, these products are biased to drift. Drift can mean a lot of things in this context. The best example to my mind is you establish a weather station next to a seedling tree. Over the next 20 to 30 years, that tree will grow, shading that station, leading to a decrease in measured temperature, which goes counter to the actual pattern that we observe with climate change. Those kinds of drifts are usually not accounted for in these gridded observation products. In addition, these products come with a lot of uncertainty that is usually not reported, giving you the false sense of 100% certainty. These uncertainties correspond to conversion from satellite data, if satellite data is being used, homogenization processes when different loggers are used at different weather stations, which is always the case, particularly when running this at global scales, and interpolation methods, which sometimes, and I'll get to this in just a second, are not very well suited for interpolation of certain climate variables. Ultimately, then, we end up with observation products, these gridded observation products, that are limited in coverage, subject to bias, have large uncertainties that go unreported, may be unreliable, and because you have limited coverage of ECVs, you have to go and look at different data sets, merge them with one another, which are inconsistent between each other. Meaning that you have to work with a fruit basket rather than just one single item. However, I would like to quickly talk about limits of interpolation, because this is what gridded observation products like WorldClim, which most of you have worked with so far, I assume, are built around. The most important thing that I want to highlight right away is that you should never statistically, which is what those methods do, downscale or interpolate precipitation data. You should also not trust anyone who does. That includes WorldClim. If any of you use WorldClim after my presentation today, I have failed. It was a great product for its time. It is still a great product for you to do explorative analysis with. I would strongly caution you against using it for anything but explorative analyses, and definitely not for any published manuscripts or research articles. The reason being that what WorldClim does, and I'll take a quick tangent here because this is a very strong statement, and I'm not coming for the authors of WorldClim itself here, but I want you to understand why I have such beef with it. WorldClim interpolates precipitation data using the information of different weather stations and the distance between them, as well as elevation and distance to coast as a covariate to drive patterns of precipitation data. That is not enough to understand precipitation, because precipitation is not driven as much by topographic features like elevation or distance to coast, but by atmospheric properties, pressure, wind, radiation, you name it. For those, we do not have high-resolution covariates which we could use for interpolation, meaning we can't use the actually informative covariates for statistical interpolation, meaning we have to use non-informative covariates, meaning we shouldn't do statistical interpolation in the first place. If you need downscaled, and someone already says it, precipitation data, use mechanistic downscaled products like CICERO. CICERO does a much better job of interpolating precipitation data. My question would be, though, if you actually need precipitation data, or particularly if you study plants, if something like soil moisture might not be more relevant to you. And keep this thought in your mind, because we'll come back to this in just a second. Because right now you might say, there's no data for it, but there is. You just haven't been told about it yet. The important thing to point out here is that CICERO already uses a climate reanalysis product, which it then mechanistically downscales to finer resolutions, instead of actually starting from weather station like WorldClim does. And so let's talk about climate reanalyses. Climate reanalyses are an optimum combination of dynamical models and observations, like we said previously. The process here is to take as many observations as you can get your hands on, run a climate forecast model, and then constrain the model using data collected at the time of what you forecasted to. This is called data assimilation. It's a bit more complex than how I just described it, but for the purpose of what you need to know, that is enough. These climate reanalyses then between each other differ in what observations have been assimilated, how they've been assimilated, and the skill of the forecast model, which usually gets better over time as you learn more about the processes in the system and make a better model that fits the actual data at assimilation step better. These are inherently more robust products for our use, as they give us all climate parameters that we might be interested in. At least most of those give us all climate parameters which we might be interested in from a single product, meaning no mixing and matching needed. These are dynamically consistent, have no spatial or temporal gaps as opposed to gridded observation products, are a close match to a multitude of observations due to the assimilation process, and give us a massive advancement in temporal resolution. Gridded observation products usually give us, at best, daily aggregates, usually just monthly aggregates. Reanalyses give us hourly resolutions, which, in fairness, for a species distribution modeling approach, probably isn't very much needed. However, for other ecological procedures, is very relevant. And so here's an overview of what data is out there. This is by no means exhaustive. This is just an overview of data sets that I have run into or used in my career so far as a quantitative macroecologist. On the left-hand side, you see gridded observation products. On the right-hand side, you see global climate reanalyses. You'll see immediately that global climate reanalyses have spatial resolutions that are usually a bit coarser than the finest resolution of gridded observation products, like WorldClim right over here. However, come with much higher temporal resolutions. Gridded observation products, however, are those most frequently used in ecology. And today, I would like you to rethink that. because we need to talk about what environmental data we actually need for the 21st century research. And I, for fear of now going a bit religious on you, and I do want to indoctrinate you into my way of believing, want to tell you about the holy trinity of abiotic data in the 21st century. And I would like to posit that we need three things from our data. We need a lot of essential climate variables to better understand systems. We need the data to be highly accurate, and we need data to be telling us about Earth and its conditions at high spatial and temporal resolutions, meaning small intervals. So let's investigate what we've been using in ecology for all of those different pillars of the Trinity. As far as data accuracy and spatial resolution is concerned, you'll see in a second why I'm mixing those two here, even though they are opposing or different pillars. Large areas of gridded data sets suffer from questionable accuracy and reliability. To further hammer home the point that WorldClim is not a terrific data source to actually use as reliable information for environmental conditions, let's just have a look at the WorldClim precipitation climatology. This is annual average rainfall between the years 1970 and 2000 across North America. In this case, we're just looking at Alaska. I don't know how familiar all of you are with Alaska itself and its topography. Beautiful country. I wish it was separate from the US. There are a lot of mountains in Alaska. Mountains drive pressure systems, so-called oreographic effects. There's a particularly large mountain chain right here called the Denali Range. Beautiful, amazing mountaineering, terrific. Lots of wolves, bit sad. But there's no pattern that shows up here on the precipitation map that we find, which is strange because we know that clouds bunch up against Denali quite frequently. What we do see, however, is almost a linear gradient going from south to north, from super wet to super dry. This is because the area is very poorly sampled. And if we were to blow this figure up and focus just on Alaska, and I couldn't find a better figure than this, unfortunately. But if you peer very closely, you can see that most of the weather stations are right down here, where you have that North Pacific climate that is very rainy. And then you have two weather stations dotted right up here, where it's just very dry. And you can immediately see how just interpolating those single points of information across vast, vast areas of land can lead to just smoothing out patterns instead of actually adding information. And just one more time, I want to reiterate, do not, for the love of God, use WorldClim precipitation data. Just don't do it. No matter how easy it is to get your hands on this data, spending the extra amount to get your hands on better data is always, always worth it. And treat environmental data with as much respect as you do your biodiversity data. In addition to this information being somewhat questionable, we get no data uncertainty or quality markers. And in the absence of data uncertainty or quality markers, most users of these kinds of data usually assume 100% certainty in the data that is being given, which is obviously wrong, and please don't make that mistake. Knowing about uncertainties is always better than having a data set without uncertainties. So if you can get your hands on environmental data and biodiversity data that gives you uncertainties, like the uncertainty of coordinates and meters that we looked at in the GBIF section, or here with something that climate reanalyses do by looking at the spread of what needed to be assimilated, you're always in a better ballpark. We do get spatial resolutions of 1 by 1 kilometers with WorldClim in particular and other gridded observation products. However, it seems as though achieving these resolutions, at least as far as accuracy of the data is concerned, might be too tough for the data sources and interpolation techniques used. And so coming back to the Trinity, I would like to argue that as far as data accuracy is concerned, we do not, or at best, don't know for certain that we actually have accurate data, because for one, we know that the data is definitely biased and wrong in certain places, and even in the places where we have a lot of samples that are then interpolated between one another, such as the contiguous US, we still don't have uncertainty markers, which really can spiral us out of control. We do get pretty high spatial resolution, so I'm going to put a tick there. because even though the data might not be accurate, at least the resolution's nice, which doesn't mean much if the data is not good. So then let's look at temporal resolution. Again, this might not be so relevant for species distribution modeling. However, most gridded datasets offer subpar temporal resolution. We know that temporal variability must be accounted for in the quantification of extreme events. And if you smooth out particularly volatile times of environmental change or fluctuation, let's take the month of April, for example, which is known for fluctuating crazily in temperatures. If you just smooth that out, you will probably end up with a regular spring temperature. However, if you have had serious frost events in there that are not captured by a monthly average, you will not understand why certain plants didn't flower that year. In addition, natural processes rarely happen at neat monthly intervals. Some species, particularly short-lived ones, will start important demographic stages of their life cycle right before some month ends, and then 2 weeks into the next month they're done. Using the aggregate of the previous month and the month where they terminate their particular life cycle stage will not be very informative to you. You need higher temporal resolution information. So another loss for the Holy Trinity using gridded observation products. So now we can still bank on essential climate variables, right? There have to be a lot. Well, there aren't. The problem with gridded observation products is that they rely on what's available at the weather stations. And because weather stations aren't homogenized and definitely don't measure a lot at all locations that we have on Earth, to be a bit more conservative, the creators of these gridded observation products have reduced the set of variables they look at to mostly just air temperature and precipitation and derivatives thereof. You will have probably run into the bioclimatic variables, which you'll also see again in a second. These are standard used in species distribution modeling, but they are really no more than interminglings of temperature and precipitation. And so ultimately, using these data sets, we try to understand the complex structure that is environmental conditions through just temperature and precipitation, completely neglecting other important variables like wind, radiation, or others. Meaning that I would like to claim that the Holy Trinity of what we want from our data is fundamentally broken, and it is time for a reformation. And this is where I would like to lead you today. Abandon the golden calf of gridded observations, embrace the climate reanalyses. Why should we switch to climate reanalyses? 3 reasons that correspond to the Holy Trinity itself. First of all, they're more accurate than gridded observation products due to the good fits that we get to real-world observations given assimilation practices. We also get uncertainty metrics with climate reanalyses, meaning that we can judge how much we should rely on certain data points in space and time. As far as resolution is concerned, while we have to deal with a bit of a hit to our spatial resolution, going from 1 by 1 kilometer on the right-hand side to 9 by 9 kilometer at global scale in the middle, or even 30 by 30 kilometer at global scale on the left-hand side, depending on which climate reanalysis product we want to interlink with. We go from monthly intervals, which you can see here, to hourly ones, and you can immediately see how we get a lot more information out of those hourly records. We can still go from 9 by 9 kilometer to 1 by 1 kilometer with interpolation, just not for precipitation, at least not statistically. However, we can do this with other metrics, which I'll get to in just a second. Finally, then, climate reanalyses, and these circles are to scale, offer us way more essential climate variables than gridded observation products. And when I say essential climate variables, what I mean is parameters about environmental conditions that you cannot calculate from one another. You see that for WorldClim, I'm only showing 2, even though the 19 bioclimatic variables are in there. I do not count those because they're derived from one another. You can do the same for ERA5 land and ERA5, which are currently the world-leading climate reanalysis products at global scale, as far as accuracy, coverage of ECVs, and temporal resolution is concerned, as well as spatial resolution at global scale. But I didn't see the point in upping all of the numbers here artificially by 19, because it wouldn't change the story that I'm telling. With climate reanalyses, and this is the important distinction, you get so much more information relating to atmospheric properties, pressure, radiation, cloudiness, and soil properties like soil moisture, which if we want to study, in our case, plant distributions, soil moisture is probably much more relevant to what we want to study, particularly if we know the root depth of our plants, than precipitation. Because precipitation and the actual water availability to plants that it results in is mediated further by soil type, slope of the soil, runoff, how much water is already in the soil itself, can it take any more in? All of this is already calculated in climate reanalyses. And here's the kicker, soil moisture is pretty robust in statistical interpolation because we can actually use static covariates like soil types. I've got a whole paper where I'm demonstrating this. So if you're curious about this, feel free to either read that paper or just talk to me or ask questions about it. And so now I would like to hammer home my theses about how to use environmental data in ecology. The very first one, and don't worry, there's not going to be 95, there's just 4. The first one is to please use state-of-the-art climate reanalyses when you are in need for spatial climate data products. Try to stay away from grid observation products as much as you can. There's a caveat here, which is that obtaining these state-of-the-art climate reanalyses products takes time and effort. So if you are in a time crunch to at least set up all of your analysis pipeline, as I said previously, feel free to use gridded observation products just so you can continue on with your code. But in the background, please have a different script running with which to retrieve this information. Which then brings me to the question of how do you access environmental data, i.e., how do we walk the road to reanalyses? I've authored an R package on this. I continue to maintain this R package. I would encourage you to use it. However, I'm biased in favor of it. So if you find other ways of linking up to those data, by all means use those, and then please tell me about them so I know how to beat the competition. Why isn't everyone using reanalyses yet? 3 reasons. Accessibility, disk space, spatial resolution. Let's walk through them. To get the world-leading global climate reanalysis products from ECMWF CDS, ECMWF is a big research institution here in Europe that produces these data sets. CDS is the climate data store where they host these data products with a really nice web interface. You have to go through drop-down selection menus, which is difficult to reproduce because you can't just show people a screenshot of where you clicked on different boxes. These point-and-click adventures themselves can also be a bit frustrating to go through if you need to query a lot of data, and you can't do it in R. And if I've learned one thing as someone who does software development for ecologists, it's that ecologists are very hesitant to leave the R environment. And there's nothing wrong with that. It's just something that ecologists do. Luckily, others have already identified the need to retrieve data from CDS programmatically with the ECMWF R package. Unfortunately, to launch a download via the ECMWF R package, you kind of already need to know how the reanalysis data product is structured, which is a level of knowledge that I don't think should be expected of the people who just want to use the data, like ecologists. You shouldn't have to pick up a separate degree to just be able to use these data products. So we need download specifications that are intuitive and reproducible. I just set that aside. Next, you are used probably so far to working with environmental data by just having one big raster file that you have on some folder on your computer and you just pull it out whenever you need it. That's not going to happen with climate reanalyses. There is large spatial coverage in climate reanalyses. In the case of what we're talking about, global, we have high spatial resolution of, in our case, at the highest 9 by 9 kilometers. We have large temporal coverage, 1940 to 3 months ago, updated every day. And we have high temporal resolution every hour from 1940 to 3 months ago, updated daily. And we have lots of variables, not just 2, but up to 83 in the case of Era 5, 50 something with Era 5 land. When we put all of this together, we work with an immense amount of data. The WorldClim raster that you might be used to, I think, is about 12 gigabytes. If we calculate all of this up, we are working in the order of petabytes, which you should never want to transfer across your internet connection, let alone store somewhere on your end. This is not something that you can stream Netflix while you do it, because you will probably die of old age before the download's done. So we need to limit and aggregate reanalysis data in time and space, both at the point of accessing it with ECMW FCDS, but also for what we end up storing on our hard drives. Because oftentimes you don't need the hourly resolution. You probably just need daily aggregates, 6-hour aggregates, monthly aggregates, 2-week aggregates, climatologies of 30-year span, you name it. You don't need to store the hourly resolution. But for each of your projects, you probably want to store what is most appropriate. So you need a workflow that accesses the data and then processes it. And finally, the study resolution usually does not align with reanalysis resolution. That is not to say that with gridded observation products, the study resolution always aligns with the gridded observation product, because oftentimes you probably don't want to do an analysis at 1 by 1 kilometer resolution. It's just what you have. I would even argue that if you run global species distribution models, you probably don't want to run at a 1 by 1 kilometer resolution in the first place for uncertainty of the input data on the biodiversity front reasons, but also for computational efficiency reasons. Again, another philosophical debate to be had. If we have time left over and anyone wants to challenge me on it, I'd be happy to have an exchange. So we need to interpolate reanalysis data to required spatial resolutions. Required or desired, you name it. And so in order to resolve all of this, I'm going to save you my long description of how this R package is built, because that is a separate talk in its entirety. I just want you to know that there are 3 important functions within Krigger, the R package that I've put together, and I'm currently working on a successor to Krigger that does all of this even nicer. You do your data download via the C downloads function, where you just tell it which variable you want from which data set for which time frame. And then it just pulls it down for you. It does the temporal aggregation for you. In this case, we want a 2-year average right here. We want it in the Central European time zone for the entirety of January of the span of 1995 to 1996. Both of those years, we want the 2-year average of those two. And we do it for this extent right here, which I've specified to be just Central Norway. This is also in the documentation of the R package itself, or on the separate workshop material I have up on my website for this R package. I also walk you through some of this functionality in the workshop material that I have up on the website for this seasonal school. I know this is a lot to take in right now. The most important thing for you to know is you can interface with Arrow5 and Arrow5 Land via this. You can easily get the data products that you want. You don't have to worry about how those downloads are made. You don't have to worry about the structure of those data sets. You don't even have to worry about doing spatial limitation or temporal aggregation yourself. Krigger handles all of that for you. If beyond that, you want to interpolate the retrieved data or other data, you can then use krigging, a statistical interpolation technique that is more advanced than what WorldClim is using and gives us uncertainty flags of the interpolated product to get to a downscaled product itself. Again, this is a separate talk. Just know this workflow exists and you now know where to look for it. Which then hammers home my second thesis, which is to please use reproducible data workflows like Krigger. It doesn't have to be Krigger, just use reproducible ones to prepare climate reanalysis products for your specific study requirements. With having access to climate reanalyses like Arrow5 and Arrow5 Land, or regional reanalyses. MESC Surfex is one that comes to mind for the European region, which gives us data at 5 by 5 kilometer resolution rather than 9 by 9. You are not limited anymore to making your models adjust to what is in the environmental data. You can make the environmental data adjust to what you want to model. And please make use of this distinction. So then how do you choose the right environmental data? There are lots of different data sets out there. There are lots of different climate reanalyses out there. Which ones should you pick? I think there are 4 criteria by which to pick your environmental data. These are spatial coverage, temporal coverage, ECV provisioning, essential climate variables one more time, and resolution in time and space. I'll walk you through those in the examples of species distribution modeling, again, because this is the most frequent use case of GBIF data paired with environmental data. Firstly, climate data or environmental data that does not cover your biological data in space is of no use. So if you are interested in our particular case, Germany, you probably don't necessarily need to use a global reanalysis product, even though that's what we're doing. You might get away with a European one, which would be of higher accuracy for the European domain and come at higher spatial resolution, meaning you probably don't even need to interpolate in the first place. The reason I'm showing you the global reanalysis product is because I want you to get familiar with the global ones, because your work will most likely not be constrained to just Europe. Secondly, temporal coverage. Climate data that represents conditions after your biological data was recorded is of no use. This is a particular gripe that I have with how environmental data is used in species distribution modeling, because often you will find people aggregate climate data over the years 1970 to 2000 and then relate those averages or standard deviations to observations of, let's say, trees, for example, that have been collected in the year 1972. Meaning you use about 28 years worth of information that have never affected that particular tree at the time that it was recorded at, which breaks causality. And I'll show you in a second of how I like to get around that, but we'll get to that when we get there. Next then, as far as essential climate variables are concerned, understanding ecological processes requires knowing the relevant components. And oftentimes those aren't just precipitation and temperature, and not just bioclimatic variables. So if you have additional variables available, please use them. And if you know that you need particular ones, search for reanalysis datasets that provide those. Lastly, finer resolution leads to greater computational cost, both in space and time resolution. Sometimes you need it, sometimes you don't. If you don't need it, don't use it. To put this into context, I would like to show you two examples. Let's say we want to look at trends in global distributions of Calluna vulgaris, the common heather, my personal favorite plant. We want to do this at global spatial coverage. We want to do this temporally for past and present. We want to use ECVs, in this case bioclimatic variables, but we want to use them based on soil moisture for data availability, for water availability, not precipitation, for obvious reasons. And we want to use a temporal aggregate, i.e. a climatology, because we're running with a regular species distribution modeling convention here. And we want to do this because it's global at a 10 by 10 kilometer resolution instead of 1 by 1, just so we are able to resolve those models within less than a week. We will probably end up using the ERA5 land data because it already comes at a spatial resolution close to what we need. We can calculate bioclimatic variables from this, and it has global coverage. It also includes soil moisture. On the other hand, let's say we want to look at the distribution of Columba domestica, the pigeon, in cities across Europe. Well, we want to look at Europe. Let's say we're interested in how the pigeon population thrived after the financial crisis of 2008, and whether people had more or less bread left over to feed to pigeons in cities. We probably want to use something like, aside from socioeconomic factors in this particular instance, air temperature, humidity, and wind speed, because those really affect how birds can take flight. And we want to use a temporal aggregate, however, as fine as possible, a spatial resolution to better understand what the pigeons were up to. In this case, we would probably land on this particular data set. Climate variables for cities in Europe from 2008 to 2017. Fits perfectly, right? Almost as though I wrote the example after I found the data set. Which then brings me to my third and second to last statement of how to do the reformation. Choose your reanalysis product according to spatial coverage, temporal coverage, ECV provisioning, and spatial and temporal resolution. How do you now get data relevant for the GBIF data that we have obtained and want to use for species distribution modeling in R? Well, with Krigger, as an example, you just use the bioclim function. You tell it about what you want to base water availability on. You tell it about the time range that you want to look at, and again, the extent that you want to look at. It pulls all of the necessary data for you, does all of the necessary computation, and spits out bioclimatic variables for you. This may take some time. Krigger is clever enough that if you break the operation somewhere in the middle because you want to go home and want to take your laptop with you, it will pick up from where it left off as close as possible, because it stores a lot of temporary files that it then deletes after it's done. Because I know that this can take some time and not everyone has access to server environments where they can just let this stuff run forever. What I want you to be very mindful of, though, is to consider whether bioclimatic variables are actually the best variables for your specific study purposes. Their use is convention, not universally adequate. Bioclimatic variables themselves suffer from a lot of drawbacks. I currently have a proposal submitted with the Norwegian Research Council to come up with better variables, because there's a few areas for improvement, I think. However, I don't want to go into that because I want to sit on those research ideas myself for the time being. So watch this space. There might be something coming up if I get money. Which then finally gets us to how you can use the environmental data. You now have it. You still need to link it up with the GBIF records, and then you need to actually use it in species distribution modeling. Matching GBIF records with climate data is really easy. We've already created a spatial point object of our GBIF data previously. We have our raster files that represent the bioclimatic variables across Germany. We just run the extract function from the Terra package, and we get a data frame back that gives us the ID of each observation, as well as the value of the bioclimatic layers in the raster that we've supplied for all of the different layers, for all of the different observations. And now you have the information that you need to run species distribution models. Lastly, I've harped on about the fact that climate reanalyses and the interpolation technique that my R package uses comes with data uncertainty. You are now keenly aware that analysis inputs are not certain, not the biodiversity ones, not the climate ones either. Which means that analysis outputs are correspondingly uncertain, and we need to propagate the data uncertainty somehow, which species distribution modeling currently is doing a very poor job at. Luckily, I'm not the only one who's figured this out, and thankfully, I'm not the one who has to resolve this, because there are already some experts working on this down in Australia, whom I'm currently partnering with to leverage the Krigger pipeline and the integrated uncertainty that we can get from the reanalysis datasets and the interpolation technique to actually feed combined data uncertainty into their models, and then report the data uncertainty combined with the process uncertainty of the species distribution model into their final prediction uncertainty. And so with all of that said, please use novel climate data in new ways. And I know that I've dumped a lot of information on you right now, so I want to close this with a few key take-home messages. First one, use big data critically. Second one, please write reproducible data workflows. And thirdly, cite data and curators. And that's me.